Alright, hopefully this is all a review from biology. So we're going to talk about Punnett squares. So let's review some of the key vocabulary you need before you can do these Punnett squares. And then we'll get into what a Punnett square is. But a Punnett square is to find the genotype and phenotype of the children. So, for example, if you were talking about these genetic diseases like sickle cell, you're going to want to find out if what the percent chance of having a child with sickle cell is going to be. So you're going to want to find out the genotype, which these are the genes or the alleles that someone has, and they are shown by letters. Alright, so you might have big A, big A, or little a, little a. The phenotype is a trait. Alright, and then in our case, we're generally talking about either sickle cell or no sickle cell. However, this could be really any trait. And I spelled sickle cell wrong, but so just what is their appearance? What do they actually look like? And the genotype determines the phenotype. Alright, so there's two types of genotypes. There's something called homozygous and something called heterozygous. So remember, the genotype is the letters. Hetero means different. So letters would be big A, little a. It doesn't actually mean they're different letters because we always use the same actual letter for the gene, but they're big and little. Alright, so these would all be heterozygous. Homozygous is the same. Alright, so we're talking two capital letters or two little letters. Alright, this is also sometimes a carrier, because if this was sickle cell, this person would not have sickle cell, but they're carrying the sickle cell letter. So they could carry it and pass it down to their children. Alright, but they don't have it, so they call it a carrier. Whereas these, they either, if we're talking about sickle cell, they would not have it, they do have it, which brings us to our next thing, which is dominant and recessive. And this is our last set of definitions here. So each gene has two alleles, which we just showed in those genotypes. An allele is just a type of a gene, or a version of that gene. So everyone has a dominant and recessive allele. That's why the genotype is always shown by two letters. The dominant allele will show by a capital letter, and this one is always expressed. Alright, so if you have big A, or if you have big A, little a, both of those give you the dominant phenotype. Whereas recessive allele is the little letter, and this has to be by itself to be expressed. Alright, so the only time we'll get the recessive phenotype is if you have two little a's. So the big one, if you have a big one, it always turns out to be the dominant phenotype. So now let's look at some Punnett squares. So again, you use Punnett squares if you have parents that want to know what are the chances of them having an offspring with a certain gene or a certain trait. Alright, so this first one we have tall and short. Tall and short doesn't really work this way, but we'll use it anyway. So heterozygous, tall person. So that means tall is dominant, because heterozygous is always dominant. So we're going to use capital A, lowercase a. Homozygous recessive then is our short person. So little a. So you put one parent across the top, one parent down the side. And then you just fill in the boxes. So big A is here, so big A is going to go in both of these boxes. Little a is here, so little a is going to go in both of these boxes. And then you do the same thing across. Little a is here, so little a is going to go in both these boxes. And then little a is here, so we go right across and go in both of those boxes. So our genotype percentages, remember the genotype are the letters, so I'm going to write down the two types of genotypes. Big A, little a, and little a. And since there's 4 boxes, 4 quarters make $1, right? And each quarter is 25%. or 25 cents, these are all 25%. So if there's two of them, big A, little a, then that is 50%. Just add them up. Little a then is also going to be 50%. So this number, these two numbers added together should always add up to 100. The phenotype now is whether it's dominant or recessive. So what I do is I go and I circle all the boxes that have a capital letter. Alright, there's two of them. So that means there's two dominant, two recessive. That would be the ratio. The ratio is just a number compared to another number. And then phenotype percentage would be, again, just like our genotype, it would be tall would be 50% and short would be 50%. Because half of the boxes are little a, half of the boxes have a dominant letter. Alright, here's another example. Now we have two heterozygous black cats. So we're going to try to figure out the phenotypes and genotypes of their kittens, if they would have babies. So heterozygous, remember, is big, little. And we have two of them. So both parents are big, little. So again, we fill it out. Big A comes all the way down here. I usually always do the big A's first just to make it less confusing. So big A goes all the way across. Little a goes down. And then little a goes across. So now, the genotype percentage, I have big A, I have big A, and I have little a. Big A, there's only one of them, so that's 25. Big A, there's two of them, so there's 50. And then little a, there's one, so 25. Alright, and again, that adds up to 100. Now, to find their phenotypes, again, I circle all the boxes that have a capital A in it, which is 3. So 3 to 1. Alright, there's 3 dominant to 1 recessive. So there's 75% is going to be black. And then 25% is going to be the other color, whatever the other phenotype is. I just didn't specify in here. Alright, so I'm going to do one more, and then you guys can do the last two on your own, just to save some time in this video here. Because hopefully you're getting this, or you can just come ask me if you have questions. Alright. So we have a homozygous dominant white cow. So again, homozygous dominant, we have 2 capital letters crossed with the red cow. Well, it doesn't tell us what the red is, but since white is dominant, red has to be recessive. So little a. So we bring our 2 big A's down, bring our 2 big A's down, bring our little a's across, and then we get our genotype percentage. The only one is that, so 100%. So all these guys are going to be big A, little a. They're all going to be carriers. And the phenotype ratio would be 4 to 0 because there's all capital A's in all of our boxes. So 100% will be white. Alright, so I'll let you guys do the next 2 on your own. And you can ask me questions if you need to. And hopefully you're getting this. And I'm going to move on to combining this with pedigrees. So those are the 2 that you can do on your own. Alright, now the pedigrees. Alright, so here's our pedigree. And here's our numbers that we're going to look at. So first we're going to look at couple 1 and 2. So what's the chances, if this is sickle cell, what are the chances that these 2 are going to have sickle cell kid? So remember, sickle cell is recessive, and all the people who are completely filled in have sickle cell. So we already know that this guy's little r, little r, little r, little r. I'm going to use r's now instead of a's. Alright, these guys are dominant, so they're capital R, capital R. And then these guys are carriers, the ones that are halfway filled in. Alright, so what's the so now we just do a Punnet square. All you do is Punnet square that with them. So we know that for 1 and 2, it's big R, and little r, little r. I'm cheating and I'm not writing. I'm going quick here. Alright, and that's what you would get. So they're all big R, little r, so there's a 0% chance of getting a sickle cell person. there's a 100% chance of getting a carrier or someone with the trait, and a 0% chance that they'll be absolutely normal. Alright, so our next couple is 3 and 4. So 3 and 4 are down here to our bottom left. And we already found their genotypes. It's big R, and big R, little r. Alright, so big R is going to be 50%. So 50% is going to be normal. And 50% are going to be carriers, or the sickle cell trait, because these two are our normal ones, these two are our carriers. Alright, and then our last one is going to be 5 and 6. So 5 and 6 is big R, little r, and little r, little r. A carrier and someone who has sickle cell. So what you see here is big R, little r, big R, little r, little r, little r, little r, little r. So we have 50% are going to be carriers, and 50% are going to have sickle cell. Alright, and that's because, and you can see that this pedigree here, down here, is actually wrong, because it has somebody who's normal down here. Right here, that can't happen. At least not with the trait we're talking about. This might be a sex-linked trait, which we're not going to talk really too much about in this class. So these guys cannot have a normal child. Alright, so that's all you need to be able to do, is really take this pedigree, be able to identify the genotypes, and then do the Punnett square to determine the possibilities for the children.